Good afternoon, friends. Last lecture we have to start the fermentation industry. Therefore, this is the second topic, that is the sugar and fermentation industry. And therefore, in the sugar and fermentation industry, therefore we have to end of the sugar industry and then we have to start the fermentation industry. In this fermentation industry, we have to discuss the introduction of the fermentation industry. Therefore, we have to discuss its importance of the fermentation, then basic requirement of fermentation, and then we have to discuss the factor favorable for the fermentation. Okay, the factor favorable for fermentation, therefore this is the 5 step, that 5 factor it is a useful for the favorable condition for the fermentation. Okay, the first factor is about the temperature. Okay, first factor is about the temperature. The temperature, it is a favorable for the fermentation. Okay, the first is temperature, that the temperature should neither too lower too high, because the enzyme destroyed at the high temperature and the inactive above. Okay, therefore this temperature, okay, in this temperature, that is the first favorable factor, that is the temperature. Therefore your temperature should neither too low or too high, means your temperature is neither too low or the neither high, means you have to take the reaction. Therefore this reaction, its temperature, you have to take the fermentation reaction. In this fermentation reaction, therefore your condition of the temperature, it should not less or not high, because therefore the enzyme is destroyed at the high temperature. Okay, your fermentation reactions temperature is high, therefore your enzyme is destroyed. Therefore the enzyme is destroyed at high temperature and the inactive above the 80 degrees Celsius. Okay, means your enzyme is at destroyed at the high temperature and it is inactive at the 80 degrees Celsius, and therefore your temperature should neither too low or too high. Okay, so this is the first point in this temperature, that temperature it is useful for the useful factor for favorable of fermentation. Okay, second point in this temperature, that is the optimum temperature range, that is the 30 to 35 degrees Celsius is the most favorable for the fermentation process. Okay, means that optimum temperature range, that is about the 30 to 35 degrees Celsius, it is a most favorable condition for the fermentation process. Okay, for fermentation process, therefore the optimum range temperature of the fermentation process is about 30 to 35 degrees Celsius. Therefore this optimum range, it is a favorable for the fermentation. Okay, that this is all about the temperature, and that is the first factor, that is about the temperature. Second is aeration. Okay, second is aeration, that the fermentation proceed only presence of air. The fermentation proceed only presence of air. Okay, therefore you have some of the fermentation process, and that fermentation process, it is a proceed only in presence of air. Presence of air means what? Therefore your fermentation process, and therefore it is a air, it is a floated air, it is passed into the fermentation process, and therefore it is a proceeding, therefore it is called as aeration. त्याला काय म्हणतो आपण? aeration. Aeration means what? की the fermentation proceed only presence of air. Only presence of air, means some of the fermentation process are proceed by the only presence of air, means the air, it is a main factor for the proceeding of the fermentation process, and therefore it is a presence of the air and it is proceeding the fermentation process, and therefore this is the second factor that is favorable for fermentation, is about the aeration. Okay, third factor is about the concentration. Third factor is about the concentration. Therefore the high concentration of this solution to an enzyme is inactive. High concentration of the solution to an enzyme is inactive. Okay, therefore some of the solution you have to taken and its concentration is high. Okay, some of the solution you have to taken into the fermentation process and its concentration is high, therefore your enzyme is inactive. Okay, so this is the third factor we are to useful for the favorable for fermentation process, that is the concentration. Okay, then fourth is presence of the other substances. Presence of the other substances, such as the inorganic solution act as a food for fermented cell. okay, the sum of the inorganic salt solution, and this salt solution, it is useful for food for permanent cell. Permanent cell means what? Therefore the different types of the fermentation process. In the fermentation process, therefore the sum of the cells are present in the fermentation process, and that cells food, okay, that cells food, it is a formation by the inorganic solution, means that inorganic solution, it is a act as a food for the fermentation cell, okay, means what? Inorganic salt solution means what? It is a food, okay, inorganic solution, it is a food for permanent cell into the fermentation process, okay, that permanent cell, it is a present into the fermentation process, and therefore you have to take the inorganic solution, and therefore it is useful for the permanent cell food, okay. So this is the fourth factor to favorable for fermentation, okay. Then fifth is pH condition. Fifth is pH condition, that the pH has the great influence on the rate of fermentation, okay. Therefore your pH great influence on the rate of fermentation, okay, means what? Therefore you have to pH range, pH is a great influence means what? Maximum you have to add the pH range into the fermentation process, and therefore your rate of fermentation is maximum, okay, rate of fermentation is maximum, means this point it is says that therefore your pH has a great influence into the fermentation process, okay, pH influence into the fermentation process is greater, and therefore your rate of fermentation is maximum, and then nextly the bacteriologist is develop the acid loving yeast, okay. In this pH condition, it is a formation of the bacteriologist, and that bacteriologist means what? It is a bacteriologist means what? Some of the scientists, it is a study of the bacteria, it is known as a bacteriologist, and that bacteriologist, it is a develop the acid loving yeast, okay. That bacteriologist, it is a develop the acid loving yeast, and that yeast you have to use in fermentation process, and this is the factor favorable for the fermentation. This 5 factor, it is a favorable for the fermentation process, okay. Therefore this factor for favorable for fermentation is over. Next point is the manufacture of the industrial alcohol, okay, manufacture of industrial alcohol, that is the ethyl alcohol, okay. Industrial alcohol means what? That is ethyl alcohol. Therefore in this industrial alcohol, the manufactured by the molasses, food grains, fruit, and the hydrocarbons, okay. We are to taking the industrial alcohol, and that industrial alcohol means what? It is a ethyl alcohol, and that ethyl alcohol we have to manufacture by the molasses, then you have to form by food grains, then you have to form by fruits, then you have to form by hydrocarbons, okay. Therefore this is the four starting material you are to producing the industrial alcohols such as the ethyl alcohol, okay. So therefore we have to first manufacturing of ethyl alcohol from molasses, okay. You have to firstly discuss the manufacture of ethyl from molasses, okay. The first is the molasses contain the 50 to 55% total sugar, of which 34 to 40% cane sugar or the sucrose, okay. So you have to know about the some of the molasses contain, okay. You have to very known about in the sugar industry, therefore the extraction of the juice, therefore the some of the total liquors, okay. Some of the total liquors means what? Therefore it is a molasses, okay. You have to know about the centrifugation process. I am saying that therefore the some of the sugar also present into the molasses, that is about the 10 to 20% sugar, it is also the present into the molasses, and therefore in this point it is says that the molasses contain the 50 to 55% total sugar. Total sugar means what? It is a glucose, sugar, or the starch contain, that is all about the total sugar, and of which 35 to 40% cane sugar or the sucrose, okay. Of which 35 to 40% cane sugar or the sucrose into this molasses, okay. So this is the first point. Then secondly, the molasses converted to the alcohol by means of yeast produces the enzyme invertase and zymes, okay. Therefore your molasses that is converted into the ethyl alcohol, and that ethyl alcohol by means of yeast, okay, that ethyl alcohol by means of yeast you have to produces the enzyme invertase enzymase, okay, enzyme invertase enzymase you have to producing the molasses is converted to the ethyl alcohol, okay, and therefore this enzyme enzymase it is a decompose the glucose and the fructose into the ethyl alcohol, okay. You have to very known about the last lecture, that is you are to saying that therefore the enzyme enzymase it is useful for the conversion of glucose to the alcohol, that is about the enzyme enzymase it is a decomposes the glucose to the ethyl alcohol and the CO2, okay. Look at this reaction, that is the sugar, that is the C12H22O11 in presence of hydrolysis reaction, therefore you have to very known about the glucose and fructose are formed, okay. This is the first reaction, okay. Then secondly we have to taking the glucose or the fructose in presence of decomposition of the enzymase and you are to producing the ethyl alcohol and the carbon dioxide, okay. So this is all about the manufacturing of ethyl alcohol from molasses, and therefore next time we have to discuss the actual conversion involved in the following steps. You have to actual conversion of the involved in following step, okay. That step you have to useful for the conversion of molasses into ethyl alcohol, okay. Thank you.